Today I'm going to talk about immune changes in autism spectrum disorder. Autism, as you know, is a very broad. Autism has very broad spectrum, but its main manifestation is behavioral defects such as social and sensory motor. It has been known for many years that the patient has some deficit in immune systems, such as infiltration of adaptive immune cells into the gut. And many more recent studies shows that immune system deficit in autism spectrum disorder is prominent, and many models use immune activation to mimic the disease in animal models. Gut is a second brain or second immune system. It harbors a lot of metabolites, which is critical for the development of immune cells. As you know, there are a lot of bacteria from the oral cavity to the gut, mostly enriched in the gut. They digest polysaccharide and provide a lot of metabolites, which is critical for the development of immune cells educated in the gut. And in addition to the bacteria in the gut, there are more genetic heterogeneity by virus and fungus and other components, which add very high heterogeneity genetic and chemical environment in the gut. The gut system is evolutionary intervened with the other systems such as brain, and we call it gut-brain intervened axis. And there are a lot of recent studies that shows that the gut system really affects the brain function, and it causes some deficits in the brain function, such as autism and neurodegenerative diseases. Social behavior, which is manifested in autism spectrum disorder patients, is governed by brain regions such as prefrontal cortex and limbic systems such as hippocampus and hypothalamus, and how the gut system affects the brain function. There are many theories and studied pathways that is known, and my main interest is the gut system and the microbiota in the gut system provide a lot of metabolites, which is critical for the immune cells, and the immune cells may mediate the brain function during development of brain. The main sites of the immune and nervous system interaction is called the blood-brain barrier, which is meninges covering the brain outside of the parenchyma, and the choroid plexus, which provides CSF into the ventricle. These two main immune interface is critical for the brain development during the pregnancy and just after birth of the baby. And what I was focusing is the non-essential amino acid, which is one of the metabolites generated from the gut system. It's non-essential amino acid and should be provided by the diets, and the bacteria in the gut system may provide metabolites such as L-serine, which is my main interest. L-serine is a pretty well-known precursor for one-carbon metabolism. It generates SAM, which is like ISAM in the middle of ISAM, SAM, S-adenosyl methionine. L-serine go through the folate cycle and methionine cycle and provide methyl group to DNA or proteins such as histone. And the addition of a methyl group to this genetic material is required for the development of cells and differentiation of the immune system. The story began like 4 years ago. A clinician visited me that she found an autism spectrum disorder patient when they treated with high-dose L-serine. Suddenly they noticed their mom and speak to their mom, hi, and they change their faces with smiling and they show very improved social interaction. And in collaboration with Gyeongbuk University Hospital, and we did some small-scale clinical trial, and we recruited ASD patients and treated the patient with L-serine for 3 months and checked their indexes for ASD. It's very surprising that they all show very improved social, improved domains of social and sensory and motor functions. Now we are doing the second phase clinical trial with the same L-serine. Then how L-serine treatment changed the behavior of autism spectrum disorder patients? Because L-serine cannot penetrate the blood-brain barrier and directly affect the brain cells, there should be some messenger mediating the L-serine effect to the brain system. I wondered some immune cells may get changed by L-serine treatment, and the interaction of the nervous system and immune may drastically improve the autism spectrum disorder patients. So we first checked the patient's blood, two different approaches. One is the proteomic analysis of the patient's blood. When we collect the small extracellular vesicles from the patient blood and plasma, they show very similar distribution of vesicular sizes. And the marker expression of small extracellular vesicles such as CD63 and LX, it shows that CD63, which is our main interest, shows a little bit difference in the patient after treatment with L-serine. And we checked the proteomic landscape between and after treatment with L-serine, and we noticed that there are a lot of proteins actually dampened by L-serine treatment. Most of the differentially expressed protein were reduced by the treatment. One interesting upregulated protein was transferrin receptor, which is critical for the development of T cell. Next, we collect the immune cells in the patient blood, and we trap the single cell into a capsule and tag the messenger RNA with barcode and sequence the individual cells transcriptome. First, we found that the treatment of L-serine actually reduced some inflammatory signature, such as interleukin 12A, 12B, but as expected, we didn't see drastic changes in inflammatory signatures. Instead, immune cell proportion showed some decreased NK cells and monocyte and some increased CD4 T cells. So we wonder, L-serine may regulate the development of immune cells in the ASD patients. So we actually checked every immune cells, but most interesting thing was found from CD4 T cells. ASD patients showed reduced CD4 T cells, and one thing induced the CD4 T cells. One thing that's very prominent were the increased population of naive CD4 T cells. CD4 T cell should be educated in the thymus gland, and the sample that we collected were blood plasma, so whole blood. So in the autistic patient, the naive CD4 T cells were actually circulating a lot in the blood. in comparison to the ASD patient blood, the inflammatory disease, so-called Kawasaki disease, showed no difference in the CD4 T cell subpopulation, such as naive CD4 T cell and regulatory CD4 T cell. So the defective increase of naive CD4 T cell in ASD patient may be very specific to the disease. We further analyzed the single-cell transcriptome and found that there are two different populations of naive CD4 T cells in the blood. One is fate-determined, and the other is actually not able to differentiate normally in the patient. So the non-fate-determined naive CD4 cells occupy a lot of the CD4 cell population in ASD patient, and L-serine treatment actually promoted the differentiation of the naive CD4 T cells. This one shows the patient immune cells profile after treatment with L-serine for 3 months. Actually, it reduced the CD4 T cells, which was induced by the differentiation of proliferating naive CD4 T cells. As we saw in the proteomic profile in the patient blood, the transcriptome actually shows that the transferrin receptor after treatment with L-serine showed the increased expression in the CD4 T cells. So, and the transcriptome signature of ASD risk genes actually were almost reduced by the treatment. We believe these changes may occur through the one-carbon metabolism in the CD4 T cell development, and when we analyze the differentially expressed gene in the blood CD4 T cells, we show that we actually noticed that biosynthetic pathways such as amide biosynthesis and peptide biosynthesis processes were regulated by L-serine treatment. This clinical study was almost done by single-cell transcriptomic analysis using blood cells, but we cannot access the brain cells in human studies. So next we wanted to check how immune cells affect the brain in ASD patient. To do so, we used some animal model. When we induced immune activation during pregnancy, the offspring, they show deficits in social interaction and sensory motor behavior, such as acoustic stutter, which is a sensory response, and some short-term memory and social dominance. They show defective behavior after birth for a very long time. So using this model, we wanted to correlate the immune interaction with brain cells. The main manifestation of ASD patient is the social interaction. This animal model also shows deficits in sociability and social novelty tests. First, we checked some inflammatory markers, and we found that a very strong increase of interleukin-1 beta was sustained in the offspring for 2 months or 4 months, even after 6 months in the brain. Because L-serine also affected the immune cell differentiation, so the immune cell could interact with the nervous system across the blood-brain barrier. So first we checked changes in blood-brain barrier in this maternal inflammation-induced model. Proteomic characterization of this blood-brain barrier in mouse brain shows that impairment of the integrity of the BBB and some transport deficits in proteomic level. When we check the markers for BBB integrity, such as ZO1, it's reduced by MIA, maternal immune activations, which indicates the impairment of BBB in this model. Actually, this result were we got this result before COVID pandemic, but after COVID, there were a lot of papers linking the blood-brain barrier and the brain disorder. So it's pretty well known that the viral infection or immune activation could impair the BBB system. We did two approaches. One is single cell analysis of brain cells, and the other is analysis of immune cells in the peripheral system. You see the single cell transcriptome analysis shows a drastic increase of inflammatory signature in the neuron. And when we analyze the immune cells in the blood system using single cell RNA-seq, there are a lot of changes with the immune activation in the immune cells. Most noticeable were changes in CD4 T cell differentiation, such as CD4 T cell memory cells and regulatory cells in terms of signaling pathways. This heat map shows the changes in the signaling pathway in each peripheral immune cells, and some cells such as B cell shows increased the signaling pathway by maternal immune activation, and most of the immune cells, including CD4 T cells, show the decreased signaling pathway with the immune activation. This right panel shows the most drastic changes with the maternal immune activation. And interestingly, most of the genes noticeable from this panel indicates that the integrity of blood-brain barrier is actually compromised. ICAM1 and VWF and some extracellular matrix proteins, they mostly indicate the vascular inflammation or deficits in the integrity of BBB. So we next checked whether the BBB system in the brain is affected by a cellular component of the BBB. We focused on the pericytes, which cover the capillary of the brain system. When we check the important pericyte markers such as caldesmon and myofluorin, we noticed that actually MIA induced the total amount of these pericyte markers in the whole system, but in the BBB, the markers were drastically reduced, which means that the pericyte, which is critical component of BBB, could be affected by maternal immune activation. So we next check that when we knock down the marker gene caldesmon-1 in pericyte cell line, the changes of proteome in the cell lysate and extracellular vesicles shows that one carbon metabolism and some molecular transport system were affected by knockdown of caldesmon. So maternal immune activation could affect immune cells, and the immune cell modification secondary affect the BBB through the pericytes or mural cells in the BBB system. So we next checked whether inhibition of caldes-1 in mice model could affect the nervous system directly. So using the fluorescence reporter mice, which express YFP in excitatory neuron, when we knock down caldes-1 in parasites specifically using AAV, we noticed that a lot of fluorescence reporter positive neurons were decreased and synapse molecules were drastically changed, and the inflammatory signature, which is marked by CD11B and GFAP, showed a drastic increase by the caldes-1 knockdown in mouse system. So in summary, actually, I provided two different two different approaches using, one is the human case study and the other is the animal model using immune activation during pregnancy. ASD patient, they have a brain disorder. I provide some possibility that immaturity of CD4 T cell may be involved in this disease. Then how? Immune cell may affect the development of BBB or the integrity of the BBB, which is critical for the proper brain function. And this CD4 T cell may promote the vascular inflammation through the one component of the BBB, so-called the pericytes. And CD4 T cell and pericyte interaction may be critical for ASD, and we believe that targeting this interaction may be important to develop some therapeutics of the patients. This is my last slide, and this is my last slide. Thank you for the support for our study, and we did this first part of the study in collaboration with the company which develops some therapeutics for ASD patients, and they are members of my lab who focused on single-cell RNA-seq and proteomics analysis. So thank you for your kind attendance.